Welcome to our lecture on parasitology. Today, we're going to explore a truly fascinating and somewhat terrifying organism known as Trichinella spiralis. Imagine eating a delicious piece of undercooked wild bear meat, completely unaware that microscopic worms are hiding deep inside the muscle fibers, waiting like tiny Trojan horses. In this lesson, we will uncover how this parasite turns its host's body into a permanent home, and we will learn how to mathematically calculate the severity of the infection it causes. Let us dive into the microscopic world of this incredible roundworm. To begin our journey, we must first understand what exactly we are dealing with. Trichinella spiralis is a microscopic parasitic nematode, which is a type of roundworm. It is the primary culprit behind a severe zoonotic disease called trichinellosis or trichinosis. What makes this parasite so incredibly successful is its stealth. It is transmitted when a predator, or a human, consumes undercooked meat that contains encysted larvae. These larvae are tucked away safely inside the muscle fibers of the meat, completely invisible to the naked eye. When the infected meat reaches the stomach, the host's digestive acids dissolve the protective cyst walls, setting the parasites free to begin their invasion. Now that we know how the parasite enters the body, let us look at its highly unusual life cycle. In the world of parasitology, most parasites require multiple different host species to complete their development. However, Trichinella spiralis is a remarkable exception. It uses the exact same individual animal as both its definitive host and its intermediate host. The definitive phase, where the adult worms mature and reproduce, takes place in the host's intestines. Then the intermediate phase, where the new larvae develop and wait for the next host, occurs right inside the very same host's muscle tissue. This brilliant evolutionary adaptation ensures the parasite never has to leave the host's body to survive. Understanding this dual host strategy brings us to the actual journey the parasite takes inside the body. Once the larvae are freed in the small intestine, they quickly mature into adults and begin to mate. The female worms then release thousands of live, newborn larvae directly into the host's tissues. These tiny newborns enter the bloodstream, using it as a highway to travel throughout the entire body. Their ultimate destination is the striated skeletal muscle cells. Once they invade a muscle cell, they coil up and manipulate the host cell to form a protective cyst around them, known as a nurse cell. Here, they can remain viable for years, patiently waiting for the host to be eaten. Let us visualize this incredible journey we just discussed by looking at this diagram. Starting at the top, we see the cycle begins with the ingestion of encysted meat. Following the arrow to the right, the meat travels to the stomach and small intestine, where the larvae are released. Moving down, we see the intestinal lining, where these larvae mature into adult worms and reproduce. The cycle continues along the bottom arrow, showing the newborn larvae migrating through a blood vessel. Finally, on the left, we see the end result, a larva tightly coiled inside a striated muscle fiber, fully encysted and ready to start the cycle all over again when consumed by the next predator. Having seen how the parasite spreads through the body, we must now ask, how do we measure the damage? To understand how sick a patient will get, doctors and parasitologists must calculate the infection intensity, which is commonly referred to as the larval burden. The severity of the disease, trichinosis, is directly proportional to the number of larvae that have successfully encysted in the host's muscle tissue. By determining the concentration of the parasite in a patient biopsy, medical professionals can gauge the sheer scale of the invasion. This mathematical calculation is absolutely vital because it dictates how aggressive the medical treatment needs to be to save the patient. This need to measure the disease severity leads us directly to the infection intensity formula. The formula is I equals L divided by M. Conceptually, this is a simple density calculation. I represents the infection intensity, which tells us the concentration of the parasite, measured in larvae per gram of tissue. L stands for the total number of larvae actually counted under a microscope. Finally, M represents the mass of the muscle tissue sample, measured in grams. If the number of larvae, L, increases while the tissue mass stays the same, the infection intensity goes up, indicating a much more severe infection. Conversely, finding the same number of larvae in a larger mass means the overall intensity is lower. Now that we have our formula, let us apply it to a real-world medical scenario. A patient is admitted with severe muscle pain, and a doctor takes a small muscle biopsy. We are given that the mass of the biopsy, M, is 0.5 grams. The laboratory technician counts exactly 150 larvae in this sample, so L equals 150. We are asked to find the infection intensity. We choose our formula, I equals L divided by M, because it standardizes the count to a single gram. We substitute 150 for L and 0.5 for M. Dividing by a half is the same as multiplying by 2, which gives us our final answer of 300 larvae per gram. In clinical terms, anything over 100 is considered a heavy, life-threatening infection, explaining the patient's severe symptoms. While the larval burden tells us the current state of the muscles, we must also look at the source of the invasion in the intestines. Once the adult worms establish themselves, the females begin producing live offspring at an alarming rate. To understand the sheer scale of this parasitic invasion, we estimate the total systemic larval load. This represents the total number of new larvae migrating through the host's body. By looking at the number of female worms, their reproductive output, and how many larvae survive the host's immune system, we can predict the future severity of the disease. This concept perfectly demonstrates why even a very small initial ingestion of infected meat can rapidly explode into an overwhelming systemic infection. To quantify this massive reproductive threat, we use the total systemic load formula. The equation is N equals W times F times S. Conceptually, we are calculating the total production of offspring and then subtracting those killed by the immune system. N represents the total systemic larval load, which is the final number of larvae that successfully encyst. W is the initial number of adult female worms in the intestine. F represents fecundity, or the average number of larvae produced by a single female. Finally, S is the survival rate of these larvae, which must be expressed as a decimal. If the initial number of worms increases, or if a weak immune system causes the survival rate to increase, the total parasitic burden will rise dramatically. Let us put this formula into practice to see how quickly the parasite multiplies. A person ingests meat, resulting in 200 adult female worms, so W equals 200. Each female produces an average of 1,500 larvae, making F equal to 1,500. The host's immune response allows only 40% to survive. We are asked to find the total systemic larval load. First, we must convert the 40% survival rate into a decimal by dividing by 100, giving us an S value of 0.40. Next, we multiply the 200 worms by their fecundity of 1,500, revealing a massive gross production of 300,000 newborn larvae. Finally, we multiply this by the 0.40 survival rate. Our final answer is 120,000 successfully encysted larvae, showing how a small infection causes widespread muscle damage. Seeing those staggering numbers brings us to a crucial realization about public health. Trichinella spiralis is a highly adapted parasite that relies entirely on predators eating infected muscle tissue to continue its existence. Because it hides so well within the meat, human intervention is absolutely required to stop its transmission. This is exactly why modern agricultural safety standards and meat inspection laws were established. By thoroughly cooking pork and wild game, the intense heat destroys the encysted larvae before they can ever reach our stomachs. Similarly, freezing meat at specific temperatures for a set duration effectively breaks the parasite's life cycle, keeping our food supply safe from this hidden threat. We have covered a lot of ground today, so let us review the most important ideas. First, we learned that Trichinella spiralis is unique because it uses the exact same host for both its intestinal reproductive phase and its muscular encystment phase. Second, we saw how doctors measure the severity of an infection using the larval burden formula, dividing the larvae count by the tissue mass. Third, we explored the terrifying reproductive scale of the parasite by calculating the total systemic load. Finally, we learned that despite its evolutionary brilliance, we can easily defeat this parasite through proper cooking and freezing techniques. You now have a complete biological and mathematical understanding of this incredible organism. Great job following along.